Okay, this is another kind of biomolecules. Again, this is a macromolecules or a polymer constructed from a smaller building blocks. Okay? The building blocks of the proteins is called amino acids. Okay? So after we have amino acids, they connect together by condensation, and we have the polymer, which is actually the polyamide. This particular polyamide is called polypeptide. That I will talk about it later. But from now, I want you to know the structure of the building block or the alpha amino acid first. So the name alpha amino acid means that we have amino group attached onto the alpha carbon of the carboxylic acid. So as you can see here, we have carboxylic acid with kind of long chain R group. Okay? And then the amino group NH2 is attached on here, the first carbon next to the carboxylic acid, which is called the alpha carbon. So we call this as the alpha amino acid. Okay. So alpha amino acids, in nature, we have several alpha amino acids in proteins. It can be up to about 500 alpha amino acids. But when you study biology in your high school, we already talk about 20 amino acids. Those 20 amino acids is called proteinogenic amino acids, or amino acid to generate the proteins, things like that. So actually 20 amino acids is the amino acids which can be decoded by the RNA structure, by the RNA system. As you may know, when we synthesize the proteins in our cells, the amino acid is attached on top of the transfer RNA, and the transfer RNA attached with the messenger RNA, which is decoded from the DNA. So starting from DNA, by RNA transcription, we make the messenger RNA, and then by translation, the messenger RNA is used to synthesize the proteins. Okay, so the transfer RNA, which will attached on top of the messenger RNA. The transfer RNA here attached with just about 20 types of amino acids. But after you condensation, making long chain of proteins, the amino acids can be derived by other chemical reactions. So at the end, in nature, we have roughly about 500 types of amino acid. Okay? But anyway, our DNA, our transfer RNA, they locked with only about 20 amino acids. That is why we learned about 20 amino acids in our high school. Anyway, if you look at the central carbon here, this is stereogenic carbon or chiral carbon, chiral center. As you can see, we have all four different kind of chemical bondings together with this carbon atom, right? Amino group, carboxylic group, H group, and R group. Okay, so from this, if I just using the same kind of method in your sugar chemistry, okay, we just turn the molecules up a bit. So instead of drawing things like this, I have you the model here. This is carboxylic acid on the right hand. Carbon with the double bond oxygen and then OH around here. As you can see, amino group is the blue. NH2, I used the blue color instead of nitrogen. No, this is the blue color. And hydrogen is white color. R group is green color over here. So from this, by the same law of the Fischer projection, I will draw carbon, carbon, and carbon in vertical line. And as you may remember, that the vertical line in Fischer projection have to point downward, right? And the horizontal line pointing upward toward yourselves. So what you can see from this picture, okay, I just turn it up like this. So in this structure, your NH2 pointing towards your left-hand side, and the hydrogen bond, hydrogen atom pointing towards your right hand. And then we just project it on the whiteboard into two dimension right? So we make a cross here, and yeah, by the law, the cross here meaning that the vertical line pointing downward and the horizontal line pointing upward. So this is it. Okay. So in feature projection, the NH2 is pointing towards the left hand. So we call this as L amino acid. Okay. And this is predominant in nature. Somehow we can see some of the amino acids, special type of amino acid that NH2 pointing toward the right hand, and this is called D amino acid, which is very simple. Is similar to the carbohydrate system. That the OH to the right is D sugar, the OH to the left is L sugar. So in amino acid it's the same thing. If the NH pointing towards the left hand, we call it as L amino acid. So in nature, sugar chemistry, the carbohydrates, OH pointing toward the right hand is the natural abundant isomer. But for alpha amino acid, the L isomer is natural abundant amino acid. So it's come from the opposite direction with the carbohydrate chemistry. Okay, after we have the amino acid, then the amino acid can be connected, okay, by condensation using messenger RNA, transfer RNA, and ribosomal RNA. So then, in the cells, we can constructing the proteins out of this, of amino acids. So, in the proteins, we have so many kind of chemical bonding inside. We talk about the chemical bondings that can be found in the proteins. To make a protein, starting from amino acids is not very easy. Let me show you how we can condensation of amino acids together and making a protein. So if I'm starting from two types of amino acids, the blue one and the pink one, you know that the carboxylic acid can have kind of condensation with the amino group, making amide bond, okay, as a product. So we have kind of amide as a new functionalities. Okay. So in chemical process, we can do it quite straightforward by adding acid as a catalyst and then heat it up. So the water will form and nitrogen atom attached with the carbonyl. OH here from the carbonyl and H from the amino group is cleaved out as the water. So as a result, what we have here is that we have NH2 group and the carbonyl group. Now this carbonyl group was attached directly with the nitrogen in pink color. So the second amino acid come into the molecule like this. The chemical bonding forming here is the amide bond. It's a simple amide bond. But the simple amide bond here, which is linked between two amino acids together, which have a special name for this kind of chemical bonding. Instead of call this as a amide, we call this as peptide bond. As a peptide bond. So that's it. That's it. This is the product from this kind of condensation. So imagine that if you have 1 mole of the blue and 1 mole of the pink and heat them up in the presence of acid as a catalyst. This is not the only product which can be formed. So I will let you think about how, what kind of other products can be formed by this reaction from 1 mole of blue and 1 mole of pink. Okay? So if possible, you may pause the video here and draw some other products from this condensation. Okay, if you're ready, I will continue to the other products. So in this reaction, as you may see, I start with N of pink condensed with the carbonyl of blue. But on the other hand, I can starting from N of blue and then connect with the carbonyl of pink as well. And more than that, because you just mix them up together, it's not necessary that pink have to condense with blue or blue condense with the pink. Blue can also condense with blue together, or pink can also condense with pink together as well. So instead of blue pink and start with carbonyl of pink condensed with the end of blue, you have pink blue, right? You also have blue blue and pink pink as well. But these four compounds is not the whole products possible. This kind of products, okay, as you can see, we have the peptide bond. The peptide is not just the name of the bond, but it's the name of the compound containing peptide bond as well. So the compounds containing peptide bond is called peptide compound. So the word peptide is used in two ways, the name of the bond and the name of the compound. So in this case, we have four possible products. As I said, pink, blue and pink, and then pink and blue, and then double blue, and then double pink. There are four possible products now. But actually, it's not necessary that the product is formed by the condensation between two molecules. Because the pink here, you have active carboxylic. It can then condense further with amino of pink or amino of blue. Then you have 3 units in one molecule, right? And so on. So we can construct 3 units, 4 units, 5 repeating units to endless repeating units. So that means you cannot make a proteins very easy by just chemical reactions in a flask in your laboratory. This is why we have to use DNA RNA system in order to synthesize the proteins, right? So here, the product is now called, because this is the dimer of two monomer, right? Connect together, this is the dimer. So the peptide compound, the dimer of the peptide compound is called dipeptide. So dipeptide is not meaning that you have 2 peptide bonds in one molecule, but it means that it is the dimer of the peptide compound. So the product, you have several peptides compound. As I said, pink, blue and pink, pink and blue, blue and blue, and pink and pink, right? You have 4 possible dipeptides, and more than that, you can also construct tripeptide. And then tetrapeptide, pentapeptide, oligopeptide, until you have polypeptide. So you have endless possible way, right? Starting from just 1 mole of blue and 1 mole of pink, you have endless possible structure from this kind of thing. So you mess up the thing by just heat amino acids together in a flask, okay? But in living organisms, we use RNA and DNA and controlled the order of amino acids, right, to making one particular proteins. So the system is very complex because we're starting from DNA and then transcripted, making RNA, and from RNA translated again to be DNA. So everything have to synthesize and arrange the amino acid order by order very nicely in order to making a particular protein structure. Okay, so this is the first type of things, peptide bonds, which is the majorities. Every protein contains peptide bonds. But not only peptide bonds, we also have some other kind of bonds in the proteins as well. Now, let I introduce you to the second type of the protein structure. I would call this as the modified N and C terminus. As you can see, we have peptide bonds, right? So after you have peptide bonds, I will show you some kind of oligopeptides. Let I start with first amino acid in blue, I will mark it as R1. And then the amino acid, the carbonyl group of this first amino acid then connect with the second amino acid, which is labeled as R2. And then this amino acid, right, second amino acid, you use nitrogen condensed with the carbonyl. So this carbonyl is condensed with the next amino acid number 3. So I label the R with number 3, R3. And then the next one, we have an H, and then R, and then carbonyl. Okay, I'll finish with this tetrapeptide. R1, R2, R3, and R4. Okay. So this is tetrapeptide or tetramer of peptide compounds. Okay, no? So, because we have several amino acid units, first of all, we have to know how can we name it, right? Because we have A, B, C, D, E, 1, 2, 3, and 4, right? But actually, we have 2 ends. We have 2 terminus. Right? We have 2 terminus. So we can count it as this is number 1, 2, 3, and 4. Or on the other hand, we can also count this as number 1, 2, 3, and 4. So that means we have to use some kind of rules to make everyone in the world think in the same way, right? So normally because we call this as amino acid, so we will use amino group here as the head, and carboxylic acid as the tail. So this is amino acid number 1, number 2, number 3, and then number 4, from head to tail. Okay, not 1, 2, 3, and 4. We count from head to tail. We count from amino group to the end carboxylic acid. The head is not only called as head group, we can call this as N terminus. And this is the C terminus. And then C terminus is very reactive actually, because as you know, that amino group is very active. It is a very good nucleophile, can attack with everything, can condense with everything, can act as a base, react with carboxylic acid, react with phosphoric acid and everything. And again, carboxylic acid is very reactive as well. It can react with ester, with alcohol forming ester, reacts with amine, and then condense as amide, things like that. Right? But amide, which is very inert, right, is hidden down inside the structure. So amide group is inert, amino group and carboxylic group is very reactive. So sometimes your proteins or peptides can then, instead of leaving head and tails freely like this, it captured or it modified the end terminus and C terminus with something else, right? For example, if we have like acetyl coenzyme A reacts together with this one, so we can acetylate the amino group with acetic acid. Or sometimes if we have like fatty acid. Fatty acids also contain a carboxylic acid group, right? So carboxylic acid from fatty acids can also modify the N terminus, right? So instead of having kind of this thing, we have like fatty acid come around and then modify the N terminus. So what we can have is like this. It will make you a bit confusing and cannot find where's your head of the chain, where's the tails of the chain, for example. Okay, but it's not very difficult actually. Not only N terminus can be modified, sometimes C terminus can be also modified. Like instead of leaving it in the active carboxylic form, it can condense with like alcohol. So you can end up with ester group instead, like methyl alcohol condensed with the carboxylic acid here in the end, and we have ester. Not only this, sometimes this can also condense with, for example, NH2 amino group, okay? Or you can use OH from sugar and then condensed with the proteins, right? You have glycoproteins. By condensing the sugar, making ester bond with the terminal, with the C terminus, for example. So this is the modified N and C terminus. So you can have simple amide. This is not the peptide anymore because the peptide is the amide bond linked between two amino acids. But this is the simple amide because it's linked between amino acid and just a normal amine, normal ammonia molecule. And this is also a simple amide bond because this amide bond linked between fatty acid and amino acid. This is not peptide. Okay, so that's it. This is kind of modified N and C terminus. Now, number 3. Another important linkage in the structure of proteins is this one. We call this as disulfide. Disulfide bond can be found in the protein as well, but it can be found in just some particular amino acid, and that amino acid is called cysteine. This is amino acid. Cysteine is this one. You have the methylene bridge linked between alpha carbon and the SH or thiol group. This is called the cysteine. Cysteine. Okay. The cysteine, or we can make it short, right, with the 3-letter abbreviation CYS. So cysteine, right, can undergo coupling between themselves. So if we have like 2 cysteine units, the first cysteine unit and the second cysteine unit can link together by this kind of bonding. S of the first cysteine unit linked with the S of the second cysteine unit. So you end up with this kind of thing. This chemical bonding is called the disulfide bond. Okay? So the disulfide bond is very stable. Actually, this is the preferable structure. So the equilibrium is go with very high K value, equilibrium constant. So this is the majority, this is the minority. That means if you have kind of proteins, normally if you have proteins with the S or 2 of the cysteine unit together, is normally cyclize themselves to be like this because disulfide bond is very strong. Okay. So if you count the number of electrons in the system or finding the oxidation state of the sulfur, sulfur has very high En comparing with carbon and hydrogen atom. So this sulfur can withdraw electron from both chemical bonding around it. So your sulfur here is -2 oxidation number. But this sulfur, because S and S have the same En value, right? So this sulfur can take electron from just one chemical bonding with this carbon atom and make this sulfur only -1. So as you can see, the process to the right is oxidation process and going back with the reduction process. So this is very easy because just oxygen gas in the molecule, just oxygen gas in the system, in the air, can oxidize the cysteine or the thiol to be disulfide like this. So this is very stable structure. Okay? If the proteins contain sulfur, it normally undergoes disulfide formation. Now we come to the last type of chemical bonding that can be found in the peptide, is the metal complex. Because as you may see in the structure here, we have nitrogen, we have oxygen, we have many things over here, right? And transition metal always undergoes complexation with nitrogen atoms. So it's similar to the thing when you want to kind of taste the proteins in your high school. Normally we use kind of copper reagent and call this as the biuret test. The biuret test is a complexation of the metal copper ion with nitrogen in the proteins chain. So nitrogen not only complex with the copper, sometimes it's complex with some other metal ions like iron, for example. So you know the hemoglobin, right? The hemoglobins, you have iron as the oxygen transport. Actually, it's make a complex of oxygen gas and the iron atom, iron ion. In hemoglobins, the iron was captured by the proteins. So there are many kinds of metal ion that is captured in the structure of the proteins. The capturing metal ion happens with normally nitrogen atom in the protein structure. Sometimes it's not the nitrogen here in the amide because the amide nitrogen is quite inert, right? One of the amino acids which is very labile, with it very reactive with metal ion is like this one. Oops. It is this one. Oops, may I draw it again? This is called histidine. Okay, this is called histidine. May I draw it as his? This is the R group of histidine. Okay, so if I have several amino acids linked together, making a long chain proteins, right? With some of the amino acids in the proteins, we have histidine. And maybe we have more than one histidine. We have more than one histidine, like this. Okay, like this. We have 3 histidine units. So we have a lot of electron pair on nitrogen atom. And because of the structure of this protein is curved up and constructing kind of pocket site. This pocket may fit with some kind of metal ion. For example, zinc, magnesium, iron, and so many things. Okay, so after that, we can then have kind of complex between metal ion and the ligands here, side chain of amino acid. So this is one of the examples of metal complex that can be found in your proteins. So this is the protein structure. Chemical bondings in proteins, we have several types. This is just the examples of some kind of chemical bondings that can be easily found in the proteins. After we have this kind of chemical bonding, not only chemical bonding that's very important to the structure of the proteins, but also the intermolecular interactions, especially for the hydrogen bonding. It's very important to the proteins as well. Now, we will see the effect of intermolecular interactions that can be found in the proteins. It will reflect or it will cause some kind of specific character, specific duty of particular proteins to catalyze reaction, to construct kind of connective tissue and so many things. So I will show you not only this chemical bonding, but the intermolecular interactions in the next script. So in proteins, when you have kind of proteins and each type of proteins has their specific character, has their specific duties. For example, you may heard about the name, about this name, the collagen, right? Collagen. Collagen is a kind of proteins for connect things with other things, right? We call this as the connective tissues, things like that. Collagen is a very tough kind of proteins. composed of 3 lines of proteins. This is the first chain of polypeptide. Then we have the second chain of the polypeptide labeled in pink color. And then we have the third chain of the polypeptide labeled in black color. They thread together in like helical shape, but different from the DNA double-stranded helix. This is kind of triple-stranded helix. Okay, so 3 of the polypeptides can be together like this in the shape because of hydrogen bonding. As you know, we have in proteins, we have characteristic amino group and carboxylic group that is condensed together. So after they condense together like this, what we can see is they come together in like, in repetitive unit with the well organized order. I would draw it like this. Whatever. Okay. So as you can see, we have amino acid that links together, forming a long chain kind of structure. And with this kind of long chain structure, you have H, which is available for hydrogen bonding because it is a hydrogen bond donor, a good hydrogen bond donor. So these good hydrogen bond donors come in every 2 atoms. Nitrogen, no? And then 2 atoms, and comes to NH, hydrogen bond donor. Again, 2 atoms, and then NH, a good hydrogen bond donor, right? So it's come in repetitive units, every 2 carbon, and then you have NH as the hydrogen bond donor. Not only hydrogen bond donor, you have hydrogen bond acceptor over here. Hydrogen bond acceptor over here by the electron pair of oxygen atom. Okay. So again, repetitively, the oxygen comes in every 2 atoms. You have carbonyl, 2 atoms, and then carbonyl, 2 atoms, and carbonyl again. So as you can see, the hydrogen bonding system in the molecules of proteins is very, very well organized. In every 2 atoms, you have hydrogen bond donor. And in every another 2 atoms, you have hydrogen bond acceptor. So they can link together, forming kind of intermolecular interactions. So this is the reason behind the structure of the collagen. And not only the collagens, proteins can form many fancy structure, 3-dimensional structure. And not only just 1 chain, they can form 3 chains bound together by hydrogen bonding. Or only 1 chain but bound together with hydrogen bonding, curve them up and craft it into kind of fancy shape, into a bowl shape, into a spherical shape, so many kind of fancy structure. Okay? Because the proteins have their, as I say, specific character, specific duty. For example, if you have kind of sugar connect together, like starch, no? And you need some kind of enzyme in order to cut down this starch, right? Like amylase, as you know, amylase is an enzyme to cut down amylose or short chain sugar, short chain carbohydrate. So the proteins may come like this, right? So normally the enzyme is constructed from proteins. The protein that acts as an enzyme is normally in a spherical shape structure. And you know that the sugar here has several OH group, and then you can have hydrogen bonding with this. So proteins is also have NH, has very polar group as well. So those kind of polar group can then fix the position of the carbohydrates by hydrogen bonding with the sugar unit. And after that, it can then bring the water molecules come into the place and then cut down the bond between 2 sugar molecules and digest it. So this kind of enzyme is normally come in kind of globular shape or spherical shape with kind of binding site, right, attached with the substrate molecules, and the active site that brings together two molecules that have to react with each other and then collide them together at the right position, at the right angle. So normally enzyme accelerate the chemical reactions by adjust the direction of the colliding substrates to be exact at the correct place. Okay, so this is one of the proteins. I just randomly draw it like this. What is the reason behind the proteins to have this kind of structure? As I said, one of the reasons is kind of hydrogen bonding. That is come in repetitive units, right? So hydrogen bond can form this kind of thing. They have kind of sheets of proteins that link together by hydrogen bonding, and here again you have this kind of structure, which is called the beta sheet. And also, as you can see, we have kind of spherical structure similar to this one, but this is constructed from 3 peptide chains, but this is just only 1 peptide chain. So they link together by hydrogen bonding system as well. And this structure is called overhelix. So these two structures, you may heard about it in your high school. But we also have some other intermolecular interactions or some other chemical reaction, chemical bonding here. For example, you may have what I said, already introduced to you, disulfide bond. Or sometimes if you have zinc, okay, and apparently your proteins contains kind of histidine unit, okay, or maybe just kind of amino group from lysine unit, or sometimes you may also have like carboxylate group from maybe aspartic side chain, or you may have also sulfur from methionine, for example. So there are many possibilities, or maybe you have like O- or OH group from kind of threonine or things like that. So this may complex your metal. Okay, the metal can complex with side chain of protein, several kinds of side chains of proteins. Okay, not only metal complex, you may have kind of hydrogen bonding between the R group, right? Because protein, as you know, we have several types of R group. Some of the R group is like you have carboxylic acid as a side chain, or you have like phenolic OH as a side chain, or you also have amino acid over here, carboxylic here, or like just simple alcohol over here. So all of these can constructing hydrogen bond together. So this is a side chain wrapped up together, okay, using hydrogen bonding. And sometimes side chain is very, very non-polar. So if you have non-polar side chain, basically protein is constructed in water media, because in the cells there are plenty of water molecules. 90% of the cells, more than 90% in the cells is water. So if the protein is constructed in the water, so if the side chain has no interaction with the water, let's say the side chain is aromatic ring or long chain hydrocarbon, thing like that. So it has no interactions with water, but they can interact with themselves by London force. So those kind of non-polar side chain will come together. For example, this is phenylalanine. You have kind of phenylalanine or side chain like leucine, isoleucine, thing like that. So because of the non-polarity, they just come close together, trying to hide away from aqueous environment. So we call this kind of pocket as hydrophobic pocket. Okay? And the interaction here is called hydrophobic interaction. Hydrophobic interaction. But not only this, there is another thing that's very important for the protein structure, which is the water itself. As I say, the proteins is synthesized in the aqueous media, in the cells, which is full of water. So water molecules can interact with side chain, can also interact with the amino linkage over here as well, can have hydrogen bonding with carbonyl group. The oxygen in the water can have hydrogen bonding with the amino group as well. So all the water molecules can be absorbed here in the structure of proteins. So you may have water everywhere in the protein structure. For example, if you have kind of huge molecules of the proteins and you're trying to crystallize the proteins out of the water. So it's similar to when you crystallize copper sulfate, right? For copper sulfate, when you crystallize it into blue kind of nice crystal, the copper sulfate, when you write the formula, it would be copper sulfate pentahydrate dot with 5 water molecules. But in a single molecule of proteins, which is very huge, right? So it can bear up to several hundred water molecules in 1 mole of proteins. So the proteins may dot with several hundred molecules of water. It's not just 5 or pentahydrate, 5 molecules of water in 1 mole of, I would say 5 mole of water in 1 mole of copper sulfate. But for proteins like this, 1 mole of protein may trap several hundred molecules of water inside. So the hydrated water in the crystal of protein may reach up to 200 and 300, things like that. This depends on the structure of proteins, how hydrophilic of the protein is as well. And the structure of proteins here is very important for the activity of the proteins, as I said. Okay? Now that's all about the kind of basic knowledge of the proteins that you may already know about this in your high school. Now I just want to focus the things, summarize the thing up for you. Now, we come to the discussion part. Okay, I will give you some kind of assignment, some kind of problems, okay, that you think about it as a homework, and then in the next video clip, we will talk about it in deeper in detail. What is the activity of the proteins, the properties of the proteins, things like that. Okay, now going to look at my homework first. Okay, now before we end our flipped classroom, I want to give you some assignment. What you can see here is some kind of beautiful structure of proteins. Okay. For the first one, second one, and third one, these 3 oligopeptides composed of several amino acids connect together. What I want you to do is, I want you to find that how many amino acids included in each particular oligopeptides here. The first one is called matrixyl. This is using in a cosmetic cream. So what I want you to find is the number of amino acids, how many amino acids, where is the peptide bonds. Okay, so identify every single peptide bonds in the molecule. And also find N and C terminus, whereas the N terminus amino group, whereas the C terminus carboxylic acid group. Okay, and do this kind of thing for 3 molecules here. This is matrixyl in cosmetic. This is TRF, which is a kind of hormones. And lastly, on my right hand here is the bovine oxytocin, another hormones found in kind of cows and buffalo. This is called bovine oxytocin. This one come in a kind of cyclic structure. So find how many amino acids inside, where is the peptide bonds, and where is the N terminus and C terminus. So these are three compounds that I want you to just kind of investigate into the structure. Now, amino acids or proteins that may come into actions in this particular compound. The blue color labels amino acids in the proteins. So actually we have very huge proteins linked all these blue amino acids together. Okay. This is a part of a huge proteins called ATP synthase. So I shall name some kind of thing here, no? So this is matrixyl. This is kind of thyrotropin releasing factor, kind of hormones, small molecules. And this is bovine oxytocin. It's a kind of hormones as well. So this one, the blue color, is a proteins which can act as enzyme called ATP synthase. So the duties of this ATP synthase is to synthesize the protein, the ATP. As you can see here, this is the structure of ADP, adenosine but diphosphate. What you have learned from the topic of nucleic acid? No, we have adenine unit, which is the nucleobase, linked together with the ribose sugar with this one, glycosidic bond. And then your ribose sugar is normally condensed with phosphoric acid, and you have AMP, adenosine monophosphate. But now you have two phosphate group, which is called adenosine diphosphate. And this enzyme trying to connect or condense this extra phosphate group, the third phosphate group, into this whole molecules. So what the duty of the enzyme have to do is connect these two together. So what I want to ask you is, where is the active site of the enzyme? So you may heard about active site and binding site in your biology. Now you can see the actual structure of the proteins. So you have to find the active site and binding site and try to discuss how this thing can, how the blue molecules of ATP synthase can capture the pink molecules of ADP and phosphate together, and how they can kind of link them together and at the end forming the ATP. Okay, so this is the thing that we will discuss in the next video. Okay, so have fun with the whole beautiful structures over here, and we'll see you again in the next clip.